I'm going to talk about plant-based, and it's actually going to be focused on plant-based dairy alternatives. Well, I'm not going to discuss myself very long. I have a long-standing background in food microbiology, worked in different places. For the last 20 years at NIZO in the Netherlands. So NIZO comes from a dairy heritage. We are a contract research company. But the last, I would say 10 plus years, we've been diverging from dairy. We do work for the entire food industry, not just dairy, but also plant-based, and these days in precision fermentation, so alternative proteins, for instance. But today I'm actually sharing on plant-based risks and how to improve your product and your processes. Just a few additional words on NIZO. So, I mean, I'm in the area of food safety and quality, but our work is really in the domain of making new products. We work in different levels for companies at different levels of consumer expectation. And if you break it down, it's really based on trust, on product performance. So, how is a product perceived? And nutrition and health, in that domain we work, and society, and this maybe, for instance, be better processing, sustainability. Now, if we look at trust, obviously food safety and shelf life, they really go in this domain. And this is, for consumers, it's not even a thing to think about. The product must be safe, otherwise it would never be on the market. The next one is actually much more apparent for companies when they work on this. So this is the product performance. You buy a new plant-based product, you don't like it, you'll never touch it again. Done. So this is a domain we work on a lot. So we have sensory panels, we have people who know all about the product performance. And in plant-based, there are a couple of major issues. One is the beanie nutty tastes. The other one is the astringency, so the mouthfeel. So we have a lot of expertise in this domain. And then obviously the other area that we work on is health benefits. So if you now have these alternative proteins or alternative products, what does that mean in terms of health? So having said that, I'm actually going to go to the plant-based products. I'm just going to stand, well, I think I'm good here. Now, when we look at this, it's really quite different from dairy. We see an uptick in plant-based, although it's been quite stable and the businesses are struggling. We see a rapid growth in this market. But what you also see in the plant-based domain is a very short product life cycle from research, like what are we going to do, until putting it on the market, if you compare that with the traditional processes and products. Oh, excuse me. Another thing that is really very different from dairy is the diversity of all the different ingredients that you can be dealing with. So it cannot only be different ingredients in terms of like a pea or oat or all these things, but it's also the quality of what you're using. It can be an isolate, which is something that went through an evaporator and a dryer, or it can be concentrated protein, or a concentrate, which is just by fractionating dry ingredients, let's say. It can be syrups, etc. So there's all these variations per ingredient already, and not to mention all the different qualities. I learned in this trajectory that rice has 1,000 different varieties. Well, I never knew. But so all these things have impact on the products that are being developed. And then the microbial content, also hugely variable. And I will show you some information on this. But we didn't really know what was there. And what I noticed myself, so we've been working in this domain for a long time. So the product developers, I just call it the stirring and the mixing. They make something great, and then they're happy, and then they come around. It's like, can we taste this? It's like, I don't know. What did you do? How did you make it? Because I'm thinking microbiology, can there be any contaminants in there? Is it safe? How do you store it? How do you heat it? So this is where microbiology comes in, because with all these new ingredients, you may be introducing new risks. Now, if we look at dairy, we know a lot about dairy. Dairy is yeah, it's been researched for decades. We know it's fairly consistent all around the world, like in terms of contaminants and yeah, the way it's processed. We know how to deal with the different microbial contaminants in different product categories. We know that the pasteurization will kill vegetative cells. If you want to get rid of spores, you can have additional bacterification steps. So we know quite well how to deal with dairy to make drinks. For yogurt, same thing. We have great cultures. You know how to, with all this diversity, and you can really get the product characteristics that you want. Same for cheese. It's a huge heritage there. But then you go to plant-based. Mostly question marks. So what do we know? What are the hazards? What is in there? Depending on what's in there, what is your processing going to do? How is your product formulation going to stop growth? So all these questions we didn't really have an answer to. So that's something, yeah, we started working on quite a while ago. And, well, it's not fictional that there may be issues with it. So these are a couple of examples. There was a Bacillus cereus outbreak in Oatly in drink. We've seen a botulism case in almond drink in Australia. There has been very recent, well, this year, a listeria outbreak in drinks. And a couple of years ago, there was a listeria case in plant-based cheese. So, I mean, obviously, the risks are there. Now, how do you better control these risks in plant-based? So it starts with knowing what's there. So actually the hazard identification. So what type of microbes are there? What are the levels that you may expect or that you will find? And then the characterization. So what do we know about these different hazards? How do we inactivate them? How do we stop their growth? How do we prevent toxin formation? These kind of questions. So this is actually the domain we've been focusing on in the risk assessments. And another thing is that there are a lot of predictive models. Martin already mentioned predictive models, but for plant-based, there are not so many. Well, the question is then, can we use predictive models that are already available for these type of products? So that's what we started working on. So we had a large consortium with 11 quite major companies working in this domain. So it's 11 different companies, 3 research institutes, or knowledge institutes, I should say. And we started to work on these potential risks and with the aim to build predictive models and to better assess risks related to plant-based. So the first step we took was identifying what's in there, and then we characterized the organisms. So in terms of what do they do in terms of inactivation, what do they do in terms of growth. And with this, we have been building predictive models, and these models will actually help to define better strategies to tackle the issues. So the approach we took is just highlighted here. We focused, or this is one thing to mention in particular, for plant-based, in principle, every product undergoes a heat step when you look at the dairy alternatives. So it's the drinks, UHT, yogurts and cheeses, they also undergo heat steps, very often also needed to, for the functionality of the product. So the focus of this project was on spore formers. So first as I said, we identified what's in there, what is in different ingredients, and then we set out to, for the key organisms that we found, to look for information related to their inactivation kinetics for spores, starting out with metadata, so what is out there in literature, see where the gaps were, do we know enough about all these different bugs that we encounter, and so we did the same for growth data. So I will show you a bit more on that in a minute. And then if we encountered gaps, knowledge gaps, we actually fill these knowledge gaps. And with all this information, we've been building models, and we can use these to evaluate risk better in different product categories. Now, this is a first, we published this information, it's on the contaminants in plant-based. So anyone interested in details, I will refer you to the paper. So what we set out to do was analyze a large number of plant-based ingredients, and we looked for the spore formers in there, and we also analyzed the total viable count. So we actually looked at these different classes for 88 different ingredients. So it was the total viable count, total spore count, the heat-resistant thermophilic spores, the Bacillus cereus counts, and the SRC, so the sulfide-reducing Clostridia. We did this for all these ingredients, and I'm not going to go into detail, but what you see on the whole is that the total viable count is quite similar for most ingredients. More than 60% of ingredients is less than a log difference between total count and spore count. So this actually indicates that spores are a very major component of these kind of ingredients. We found Bacillus cereus in a lot of the samples, certainly more than certainly more than half, in relatively high numbers even sometimes. And of course SRCs, you can see it in the graph here, they were also found. But what is really very striking is that the levels, unlike milk, the levels are very variable. So if you look at the spores, you can have an ingredient, and this is all P isolates, what I'm showing here. The levels could be between 1 log and 5 logs. And yeah, the same goes for the different contaminants that you may encounter. The levels can be very variable and sometimes even quite high. Bacillus cereus we would encounter sometimes over 2 log per gram or more. We picked colonies from all these analyses we did, and we did some identifications on all of these. We did identifications on all of these on the highest dilutions of plates. And when you look at the spore formers, so the aerobic mesophilic spore formers, we encountered a lot of bacillus like Informus, Subtilis, but also even on non-selective plates, Bacillus cereus was often found. And well, then there was a whole other group of like a relatively more diverse group of bacillus found as well. For the anaerobes, quite a lot of the isolates that we obtained were Clostridium tepidum sporogenes. And as you may know, these are very similar to proteolytic Clostridium botulinum. So this just gives you a flavor of what is in there. And what we did next was to see what we know of these bugs to be able to have good models. So we collected data on the inactivation of all these spore formers, or the spores of the spore formers, and we collected data on their growth characteristics. So when you have a growth model, you assume an optimal growth rate, and depending on the conditions under which you then grow it, so pH or water activity, presence of lactate, for instance, or a certain food factor, you can multiply these optimum growth rates with these gamma factors to get a prediction for growth under the conditions that are relevant. So we collected this information in huge databases, and then we now have models for all these individual spore formers. So this is just an example of the data on heat inactivation of spores. So this on your left you see all the data for all spore formers that we retrieved from literature, and this is actually the log of the D value. So the amount of time you need at a certain temperature to reduce your spore counts by 1 log, and we collected this for Bacillus cereus, Bacillus, Clostridium, Geobacillus, and Pinobacillus, and also for the less abundant species that we encountered. And this just shows you on the right. It just shows you all the data within all this. In the gray you see all the data for all these spore formers, and that just shows the data for Bacillus subtilis. And then we know in Subtilis that there's 2 main groups, not so heat resistant and a quite heat resistant group. So that actually shows you the data that we use in our databases to calculate the spore inactivation. And we took some isolates from plant-based, and we assessed the heat resistance of these. And that actually showed that they were within that bandwidth that you may expect. So they were not outstandingly heat resistant. And that actually held true for the different organisms that we evaluated. We also looked at the cardinal parameters for growth for all these different species. So a lot of data is already available, so we collected all of this, and where we had data gaps, we then supplemented the data there to be able to make better predictions. Yeah, so this is just a list of organisms for which we collected new cardinal value parameters for temperature, pH, and lactate. And on the whole, we saw that the isolates from the plant-based, they were behaving the same as what we already know from literature. So on the whole, I think that what the conclusion is that we can use predictive models based on all that literature that is out there, supplemented with the data for unknown species. Now, how do we use this in practice? It's nice to have all these models. I mean, a model is a model. It's pointing you in a direction. It's not going to give you an exact answer. But what we typically do when we are assessing a situation on plant-based, we start just by applying these predictive models. So we start with a process. So we map the process. We know what kind of heating, what water activity, what pH, the batching times, like very detailed. It's really in very detailed assessment of what are you making and how are you making it. And with that, you can predict for, we have pathogens as well, all the pathogens in our databases, but also these spore formers. And this is just an example. So you have a process here, you have a bacillus, there is an initial level, you have a heating step, nothing happens, it's a bacillus, so the spores are not killed. And then you see growth. Here you have a listeria, you have an activation, and then later you also have growth after your heating step, for instance, if you didn't inactivate enough. But what we then do is, again, in silico, we take all our models and we start calculating. Okay, what if you now crank up your heating, not make your heating longer, but just go a few degrees up? How effective is that? What can you expect? What can you do with your product formulation? Can you go a bit down in your pH, or can you change your water activity? All these, this you can do in silico in the computer, and with that you actually get to a situation, a scenario where your risk organisms are then most likely controlled. We can then verify this. We have high throughput systems where we can make the actual products and treat them the way you would actually do it in practice. So we have a food application center, we have a pilot plant facility. So here you can then see if those organisms that were potentially of risk, if they can be controlled, but using a lot of different variations. So the best variations thereof, you then take to the next step, and that's your real production. And then you can actually start with the proper challenge testing, which is, expensive business, obviously. So this is just an example of that, all these questions we then have for related to the microbes. What are your initial levels? How are you going to be heat treating? What are your production processes? Do you use batching? What is your cooling like? How long do you do this? All these very detailed questions we need. The product characteristics, the typical storage conditions, to map it out and put that in a process flow. And then we match that with all the data we have on the microbes. So then you calculate throughout the process, you look at inactivation and growth, and then you see where your risks are. So now I will just switch actually to the main product categories for dairy alternatives, so the drinks and then the yogurts and the cheeses, just to give you a bit of a flavor of what you can expect there. So for the UHT drinks, these are pH neutral, receive a high heat treatment, but if you have a failure in your heating, or your heating is not high enough, or there's a problem with your hydration of ingredients, spores may actually survive your heating treatment. You will kill all your vegetative organisms, not a problem. So then you have two main potential issues. It's spoilage, That's very annoying. Can be very thermal-resistant spores, so especially geobacillus. They can survive UHT treatment. In principle, not a great issue in the market unless you start storing the products above 40 degrees centigrade, because then they can grow and they will cause spoilage. There can also be other organisms like Bacillus subtilis. There are strains that can survive these kind of heating steps. Another major cause probably of even UHT treated is spoilage by vegetative ones, but then we're looking at post-processing contamination. So here you can actually have spoilage in the market. Food safety risks. I mean, I mentioned the Bacillus cereus in the oat drink, Clostridium botulinum in the almond drink. So these are real risks, and it may be related to insufficient heating or post-processing contamination. So now just a case. So how can you determine now? Okay, what you have a spoiled product. That's one thing. Very often you can already see based on what organism is in there. Was it a post-processing contaminant or was it something that survived? Vegetative organisms is always a post-processing contaminant. But here we had a case of Bacillus subtilis. So we know that some subtilis produce superheat resistant spores. And the only thing you do when you do an identification is a name. Okay? Then you have a Bacillus subtilis. Where does it come from? Is it your ingredient? Is it your line? No idea. So the way we went about it is do the identification. So we knew the organism, we cultured the organism, and then we generated spore crops. And we determined the heat resistance of the spores. So you would expect, okay, UHT drink, Bacillus subtilis spores, maybe it was in the ingredients, and we know that these levels can be high. So we determined the D values and we plotted it amidst these cloud of data that we have. And it turned out it's actually a strain that is not very heat resistant. So that directly pointed at post-processing contamination, so not at your ingredients. And then a very practical solution is like, okay, you have to start looking at your process and your cleaning and your CIPs and all these things. But you don't have to worry, further heating is not going to solve the problem. So this is how you can use this kind of information and how you can solve problems related to that. Another thing in plant-based, it's not dairy, product sterility testing. So very often when you produce UHT drinks, you put a batch in accelerated shelf life testing. And then it's, you test at 30 degrees for the mesophiles, 55 degrees for the thermophiles. What is then the time that you need to incubate and to start sampling? And this time to detection is ideally ASAP, because it actually may, it may be a prerequisite for product release. So this detection level obviously depends on the method that is being used, the sensitivity, but also the growth rates of typical contaminants. And knowing more about how fast bacteria grow in plant-based drinks, for instance, it will help you to set that most optimal time for doing your analysis of the packs. And there are cases where we know that the incubation for 2-3 days is not enough. You need to go to the 4-5 really, because you're not going to catch organisms of concern that can later pop up in the market. Few words on yogurt-like products. So this is a very different category. Here in plant-based, it's typically pasteurized. It can be temperatures up to 90 degrees. So again, you kill your molds, your vegetative organisms. Spores will survive. They are there. And knowing now, based on all the work we did on the ingredients, we know that Bacillus cereus can really be there. We saw it in many samples. Over the years, I've also had people come to me. It's like, we are now working on this product. We have these pink colonies. We picked them. It's Bacillus cereus, actually, in a plant-based yogurt that we were trying to develop. But the thing about Bacillus cereus, it doesn't grow at pH 4.5. So this is a typical target pH for yogurts. So once you get to that pH, you should be fine. However, the risk is in the fermentation itself for plant-based. So here we know that the organism is actually a risk in the fermentation process. And this is just some data that I can share. So these are plant-based yogurts, oat-based and fava-based. And what we saw there, we used commercial starter cultures. We saw that if you don't have enough growth of the starter culture or fast enough acidification, so if you start, for instance, with not high enough inoculum, we saw that Bacillus cereus could grow pretty rapidly, much faster than what you would normally see in milk. And that really depended on the acidification rates of the cultures. And we used the cereulide positive strain, so it's a very stable toxin. And you actually could detect cereulide in your product. What may be even worse, and this happens as well, Bacillus cereus can grow, it can produce a toxin, and in acidic conditions, it just dies off. So you do product testing, no Bacillus cereus, toxin has been produced, very stable, so this is a potential risk. So this is something you must prevent in the processes for yogurt fermentations. Now, this is some other work that we're doing on plant-based fermentations, and this just shows you on the left side of the panels. So these are 4 different plant-based substrates. They were inoculated with, I don't know, 40, 50 different lactic acid bacterial strains. So what you see, this is just everything plonked into one picture. So this is actually the pH and time in each ingredient with the identical cultures. So you see that some ingredients, in time, you see overall like a faster decrease of pH. Other ingredients, they were not good at all. Like I think the top right here is oat. Oh, maybe I can point. So that's oat, but it didn't acidify very well. So you see that between different ingredients, you have very different acidification profiles even with the same cultures. Now, if we zoom in a little bit more, of all these strains, of course, we know which isolates they are. We had Lactiplantibacillus plantarum, Lactococcus cremoris, Lactis, and Leuconostoc in there. And then you see that some species are much better at acidifying than others, but also within a species, some strains are much better than others. So it's very critical to select the right cultures. And yeah, for this we are now also building predictive models to have good combinations of ingredients and lactic acid bacteria to drive this. Well, and it's much more complicated because some of these can also help to remove off flavors, and of course you want to have the right flavors in there, so the choice of culture is very important. But I think the choice of culture is also critical for the acidification to prevent Bacillus cereus risks. Now then a few last words on cheeses, so plant-based cheeses. Yeah, if you look on the packages, sometimes these products are it's just fat with starch and water. So yeah, the pH tends to be not very low, between 5.5. But the moment you start to add in all these plant-based ingredients, so the protein component, you're thereby also potentially introducing the spore formers again. Typically also undergoes a heat treatment, so again, get rid of your vegetative organisms, your yeast, your molds. But yeah, the target pH is quite critical. And then you have an additional post-processing potential contamination event, and that's very often the slicing and the packaging. So here you are introducing a potential risk for listeria. I showed you some information on acidification, so buffering capacity, and what we see here is that sometimes the buffering capacity is much lower than that of milk. So you need less lactic acid to get down to the same pH, but the lactic acid is critical for stopping Listeria from growing. So you're having different risk profiles here for these type of products. And another potential organism of risk here are the Clostridia. So here we're looking at outgrowth during storage as a potential risk, especially the Clostridia, Listeria. And then if you were to move to fermentation-based, plant-based cheeses, the same goes as for the yogurts, where you have potential risk of Bacillus cereus in the fermentation. So again, for the plant-based cheeses, we now also have nice systems where we can do this high throughput. We can have all sorts of variations on a theme in a 96 well kind of format, where you can actually test whether the formulation that you're developing is preventing outgrowth of critical organisms. And I think with that, I'm coming to the end. Just as a short recap. Looking at risks and strategies for control, when you look at the UHT plant-based drinks, it's very obvious that the levels can be quite variable of the spore formers, but also the types of spores that you can encounter in ingredients. Heat treatments, obviously crucial to achieve sterility. But yeah, if you do have survivors, and this may be a problem because of the way so you cannot use bacterification, so the levels may be higher to start with. There may be more fouling, there may be issues in hydration of ingredients. So yeah, there may be risks of spore formers surviving the UHT treatment. Or if you have post-processing contamination, you have actually a very high risk of outgrowth. So the inactivation models, they are crucial for determining whether the heat treatments are sufficient, but we also use them to assess, for instance, issues in the batching. So if you make a plant-based slurry, how long do you keep it before you put it over the UHT? So that's a potential risk as well. Plant-based yogurt, same thing. Variable levels of spores. They are not killed by pasteurization. pH typically is low enough to prevent any issues in the product, but here, as I mentioned, the fermentation step will be very critical. And then for the plant-based cheeses, it's in a way quite similar to the yogurts, but here potential outgrowth during storage is a big issue because the pH tends to be quite a bit higher. So here you're also looking at risks of Clostridia and Listeria. And yeah, here the growth models are not good enough yet, so a lot of testing is involved as well. So I would like to leave it at that, and I would like to thank my colleagues at NIZO, but also at Wageningen University. So they were and the Haas Green Academy. So they were the knowledge partners in this project that I discussed. There was some work on the acidification by lactic acid bacteria and plant-based. It's actually part of an EU project, so it's called EMUS. It's now coming to an end. And these were the TKI consortium partners. So with that, I would like to thank you for your attention. Any questions?
Thank you.